Hi, everybody. My name is Kevin Folta. I'm a professor and chairman of the Horticultural Sciences Department at the University of Florida. And today we were asked to do an interview about the book Maize Kernel Development and the author, Brian Larkins. We're doing this mostly for the American Society of Plant Biology and also for the Plante website. The idea being to familiarize more people with the text, but also with the work of Dr. Larkins, who is a lifelong ASPB member, a former president, and someone who's been decorated with just about every award the society offers. Dr. Larkins is now an associate vice chancellor for life sciences and at the University of Nebraska. And he holds the John F. Davidson and Marion Fuller Presidential Chair in Life Sciences at UNL. He received his bachelor's degree in education from the University of Nebraska-Lincoln, as well as his PhD, and spent 12 years at Purdue University before becoming head of plant sciences at the University of Arizona in 1988. Since then, he was named the Regents Professor and Porterfield Chair in Plant Sciences at the University of Arizona. He received the Charles Shull Award in 1983, the Dennis R. Hoagland Award in 1997, and the Stephen Hales Prize in 2013. And this among a number of other awards from the University of Nebraska-Lincoln. He's also a fellow of the American Association for the Advancement of Sciences and is a fellow of the American Society of Plant Biologists and also a member of the National Academy of Sciences. So quite a laundry list of accomplishments. Welcome, Dr. Larkins.
Thank you, Kevin. There's just one correction I need to make with your introduction, and that is that I retired from the University of Nebraska last summer. So I am now an emeritus professor from the University of Arizona and the University of Nebraska.
Okay, so I'll leave that in because the correction sounds better coming from you. So that was perfectly fine. Yeah, I meant to. You are retired and currently in Arizona, right?
That's correct.
The main reason that the American Society of Plant Biologists wanted me to have the conversation with you was around your book, Maize Kernel Development, which you edited last year. The chapters were written by experts in the field, and each one is really a nice, concise capsule. It isn't these long, deep, complicated chapters. And it's something that someone interested in maize kernel development or really any grain kernel development may be very interested in. So kind of going back to the book and really framing the conversation today, what was the original impetus to write a book on maize kernel development?
A few years ago, I was invited to go to China and present a series of seminars. And since I had retired from the University of Arizona a few years before that and no longer had a functional lab, I was sort of scrambling to figure out what kind of a research seminar I could give. And what I ended up doing was putting together a summary of what we learned over 35 years of research. And then I ended it with a number of questions that were outstanding questions that we still didn't know the answer to. And I was, pleasantly surprised by the response that I got from the students. And on the way home, I was thinking about it, and it occurred to me that most scientific reviews tend to be a documentation of what we know and how we came to know it with lots of references to it. And if you're new to a subject matter, you can go to the library and you can, over a period of time, get an idea of what's known. But it's much more difficult to learn what isn't known and what are the important questions that remain to be answered. And even better yet, how one might go about trying to answer them. So that was sort of the germ for the idea of putting a book together. And since I was going to be retiring, I thought, well, maybe that's something I could do in my retirement. But then I realized that if I was to do that, I would end up going to the library and finding out all the things that we know, and I wouldn't necessarily know the questions that we don't know. And so it seemed obvious that the best solution to this was to try to get a number of experts to write chapters on their research or their research area, and encourage them, in fact, demand of them that they not just write a real detailed literature review, but sort of give a synopsis of what we understand, but then expound upon the questions that we still don't have answers for. and how one might go about doing that. And I also thought that it wouldn't be a good idea or perhaps fair to ask a bunch of young scientists to do this because they may not be comfortable sharing some of their best research questions. And so, hence, the people that I asked for the most part were people who are well established and well known for their expertise on different topics.
And that was really one of the interesting things about this was the speculative nature of each chapter, that there were a lot of forward-thinking questions, that here are the buckets to be filled. And I think that's really a great thing for encouraging younger scientists who really look at the depth of the literature and the depth of expertise and feel, where do I find my niche? And was that kind of the idea behind it?
Yeah, that's correct. I don't know if it's the right way to say this, but when I started off as a postdoc / assistant professor, I had the luxury of starting off on a research project for which there was basically little or no literature. So I didn't have to spend a lot of time in the library trying to learn what people knew. Of course, for some subjects it's always a problem, but it's just very helpful if you can read something that helps direct you in terms of what isn't known. Even if you're not doing research on it, it still helps you think about the subject matter.
So why the maize kernel? I mean, we all know that maize is an economically important crop, but what is it about the kernel and kernel development that makes it especially appropriate topic for this particular text?
Well, you know what they say about writing, you should write about the things you know, and having spent 30 or 40 years working on maize kernel development, that was what I knew the best. And I certainly knew who the best people were to contribute to it, which is also a pretty important aspect of putting together a book like this.
And development really underlies the economic importance of the maize kernel, and that is really its constituents. And so what do we look for in a maize kernel as a consequence of development?
Well, of course, the three major things that at least the earliest research was done back in the early 80s was on the things that we use the kernel for, that is starch, oil, and protein. And those are still the most important things. I mean, the economic value of maize is price per bushel, and that's determined by the starch content. And so that's always an important aspect. But it also translates. It just so happens that all three of those components are very important for making food products, and as well as livestock feed. And so it's sort of it's obviously important not only biologically, but also from a practical and economic point of view.
And also for fuels, right? I mean, how much there's a certain amount of corn that goes into the creation of ethanol.
In the United States, that certain amount is about a third of it, at least at the present time. Of course, originally, people didn't think of corn as a way of making biofuels, but rather of making alcohol, which has other attributes.
Sure. But when you look back at how kernels and their development have changed, you look back to something like teosinte, and how has selection and breeding and 10 to 16,000 years of domestication changed the corn kernel?
Well, without getting into an awful lot of details, the first chapter of the book by Sherry Flint Garcia it gives a very nice synopsis of how we learned how maize was evolved, which it was evolved by humans from teosinte, which has a very small, very rock-hard, little tiny seed, into modern-day maize, which has a very large, flat seed that's about 10 to 15 times the size of a teosinte kernel. John Dobley and his co-workers over the years have identified I think there's thought to be about 6 major QTLs that were responsible for transitioning teosinte to maize. One of those is a transcription factor that the mutation of which causes the very hard fruit case of teosinte to not form, so that you get a soft, pliable kernel. And another one was another transcription factor that causes a plant. Teosinte is very branched like a grass. Um, and of course, modern maize has principally a single shoot. Well, anyway, both of those were quite important in terms of the morphological changes that took place from teosinte to maize to make it become the important food crop that it is today.
And so when we look at the highlights of the genes that separate them, is it really just a couple of transcription factors, or are there other really important watersheds genetically that occurred that really define the corn cob and the corn kernels, so that that funny little thing called teosinte really differentiates it from what we know today as modern corn?
Well, there are several of these key QTLs that have remained to be identified. So it's hard to guess what they might condition. But among the things that Sherry talks about in that chapter are the changes in population genetics that contributed to changes in yield and high amounts of starch and high amounts of protein and so on. So there's just not enough time to get into those details here, but if you're curious, you can get it in chapter 1.
And so that's chapter 1, but could you give us maybe some other highlights, maybe dance around the different chapters of the book, and what are some of the other topics that are covered?
The book covers everything from the fertilization process up to the point of the early developmental phases of the formation of the endosperm and the embryo, kernel filling, kernel maturation, and things that affect seed formation. The final chapter deals with the effect of drought on kernel set and kernel filling, which is a very important topic as far as yield is concerned.
And with respect to that, someone who maybe wasn't a maize aficionado or really studying maize per se, would they find this information useful maybe in translating to other grains?
Yeah, so the genome of rice was sequenced before maize, and so it quickly became a model system for cereals, and that included many of the traits associated with kernel formation. But the rice kernel is quite different than the maize kernel. It's not it has a different protein composition. The starch part is the same, but it's not a high oil seed, and of course it's much smaller than the maize kernel. So there are some subtle differences, but to a large extent the basics are similar. And the way things are today, we get as much insight about some aspects of maize kernel development as maize has provided for rice kernel development.
And is there a lot we've learned about maize kernel development that translates to issues of, say, world food security?
Well, the molecular biologists are reductionists, and they like to think that if we understood the molecular and biochemical basis of all of the processes going on in the seed, that we would be able to engineer or manipulate the seed to improve some of the deficiencies that may exist. We certainly know we've learned a great deal about a number of things, largely because of just the economic importance. And so a lot of research has been done on how starch is synthesized, on how the storage proteins are, or the nature of the storage proteins and how they're made and stored, and also about the synthesis of oil, which occurs in the embryo. So I would say there's probably more detail about those biosynthetic processes than about much of the developmental processes. But and I guess the other thing to add to that is that back in 1975, when I first started working on this project, there wasn't a lot known about the molecular biology of plants. And what attracted me to work on maize storage protein synthesis in the first place was that I learned that the protein in the maize kernel, much of it was a single type of protein, a storage protein called zein. And in that respect, it was like another model system in animals that is the synthesis of hemoglobin. The major protein made in red blood cells is hemoglobin, and it was one of the first systems in which they identified how many ribosomes there were on a messenger RNA that made hemoglobin. And I thought that the storage proteins in corn kernels might be an equivalent system for plants, and indeed it pretty much turned out to be the case.
And you spent so much of your career looking at seed storage proteins. What are some of the other important reasons to consider their synthesis in either the role of the nutrition of the kernel itself, or perhaps even in the physiology and maintaining the embryo? Or tell us a little bit more about seed storage proteins and why they're important.
Well, basically, the seed storage proteins are a way that the plant can store nitrogen and sulfur for subsequent use by the embryo when the seed germinates. The storage proteins typically tend to be, well, at least as the kernel desiccates, or as any seed desiccates, it loses water, and so the proteins become insoluble. It just so happens that in cereals there is a type of storage protein that was found to be very high in proline and glutamine. They're called prolamines, and the prolamines determine the unique, what we call the functional properties of the meal or flour produced by grinding those seeds. So, for example, the prolamine proteins in wheat determine the elastic nature of the dough, which allows you to make bread or pasta. The proteins in corn do not have the same sort of flexibility that the wheat proteins have. So you know that cornbread or corn chips are quite more brittle than what wheat products are. It just so happens there's very little of those prolamine proteins in rice or in oats. And as a consequence, well, I should say, those prolamine proteins are generally essentially devoid of several essential amino acids, lysine being the most limiting one. And so as a consequence, you have other proteins in rice and oats, and they turn out to be, as a grain goes, a more nutritional source of protein than what you get in corn or sorghum or wheat, for that matter. Anyway, so besides their importance as far as the nutrition is concerned, and maybe I should return to that very briefly, because I mentioned about lysine being an essential amino acid. In societies that rely principally on one type of cereal, and it's not uncommon that be corn or sorghum, particularly in Africa, that it's often the case that mothers, when they're weaning their children from breast milk, make a gruel of the cereal, and they feed that to the children. And the deficiency of lysine results in a disease that's known as kwashiorkor. Kwashiorkor means the sickness the first child gets when the second child is born. Anyway, it's a protein deficiency disease, and it comes about from having insufficient amounts of some essential amino acids. So to go back to your original question, this was one of the reasons that I got as interested as I did initially in working on the storage proteins in corn, because there had been a long-term aspiration of biochemists and cereal scientists to try to find ways of increasing the lysine content of a corn seed. And there are mutations in corn that bring this about, although they have side effects that are not particularly beneficial from an agronomic point of view. Anyway, the other thing that's important that I alluded to about storage proteins is that they affect the functional characteristics of the food products that are made from each cereal. And so the food products are unique for each type of cereal, largely because of the kinds of proteins that they contain, because the starch is pretty similar among them.
I guess when we looked at your record, you look through actually a publication record that goes back quite a ways in seed storage proteins. But in the last 10 years, you really focus more on cell cycle. And how does that pay into this?
So when I was a postdoc, beginning assistant professor, I read a review in the annual review of plant physiology about a process called Enery Duplication. Enery Duplication occurs in a number of plant tissues, notably in seeds, fruits, and it involves making multiple copies of the genome. So, whereas the cotyledons of a soybean plant typically would be like have a 2N complement, you can get 3 or 4 cycles of Enery duplication, so they end up having 16N or more. And at least at the time, it was thought that this was a mechanism that was used by the cell to amplify gene expression to support high levels of metabolism. So you made more starch, more protein, whatever. It's also the case that endoreduplicated nuclei tend to be associated with larger cells. And so it's a way of enlarging the tissue as well. But most of those hypotheses, I guess I should say, were not substantiated by experimental research. Without getting into the details, one of my postdocs, Jeff Haben, was working on a project where we were trying to understand why certain mutations in the endosperm of corn caused there to be a higher lysine content. And we wanted to identify the proteins that were being increased that were responsible for the high lysine content. And so what we did was to just make a complex antiserum against all of the endosperm proteins, and we used it to screen a cDNA expression library. That is a library of clones corresponding to all the messenger RNAs in the endosperm that were in a plasmid that would allow them to produce protein. So the idea was that the major proteins would be represented by the abundant antibodies, and that by identifying the sequence of the gene, we would be able to figure out what the protein was and how much lysine it contained. Anyway, so Jeff just started off sequencing these clones, and one afternoon, Tim Helen-Jarvis, who was helping on the project, came over to my office and said, one of these genes looks like a human tumor suppressor called retinoblastoma. And I had just read a book about the discovery of the retinoblastoma gene. It's a really interesting book called Natural Obsession by a woman who worked in Harvey Weinstein's lab at that time that they discovered this tumor suppressor. So anyway, it got me thinking about, wow, that's pretty cool. So plants got the same kind of genes that affect cell cycle regulation that mammals do. And I thought, well, maybe it has something to do with this endoreduplication process in the endosperm. And so that was sort of the linchpin that got us started trying to figure out how the endoreduplication cycle works, and to create mutations of the endoreduplication cycle that either have increased amounts of endoreduplication or reduced amounts of endoreduplication, and then to see what that does to the starch and protein content of the seed.
Okay, so maybe we'll see you there, at least through LinkedIn, et cetera. That's great. So one of the real benefits of the book is kind of open-ended and forward-thinking speculation in each of the chapters from the experts. And why is that important? I mean, haven't we already discovered everything?
It's amazing. I never went through to count the number of references for all the chapters in the book, but my guess is there are, somewhere about 500 to 1,000 of them. But there's amazing the gaps in a number of areas, especially as it relates to development. So, we know a lot about the biochemistry by which the storage proteins and starch and lipids are made. But, to form the seed, there has to be some kind of communication and signaling that takes place between the plant and the ear and the kernel. We know that somehow the metabolites are partitioned between the endosperm and the embryo. So the endosperm stores starch primarily as a carbon source, and the embryo stores oil. But how it partitions the oil to the embryo isn't clear. There has to be some kind of communication between the endosperm itself and the embryo. And in fact, there are mutants. Well, so if you when you eat corn and you pop the embryo out, you'll see that there's a little groove that's left behind in the endosperm where the embryo sat. There are mutants that block the formation of the embryo, and they'll still form that groove. That groove is an area of active transport of metabolites and signals between the endosperm and the embryo. And we know almost nothing about what the mechanism is and what the signals are that go back and forth between the two. For that matter, we don't know much about the signals that come from the plant that go into the kernel through what's called the basal endosperm transfer layer, which is a key cell layer by which the sugar leaves the leaf, the plant, and goes into the kernel. There's a thing that is called the kernel or the endosperm is thought to act as a so-called sink for nutrients. It accumulates amino acids and accumulates sugars to make starch and protein. What drives that transport is called the sink strength, and we know very little about what the mechanism by which sink strength is established. The other thing that people might not be aware of is that they know that corn kernels are large, but the kernel is so thick that the level of oxygen, which you need to make ATP for all the biosynthetic reactions, is very low. So how does those cells in the central part of the endosperm create the energy that is needed for the biosynthetic processes if there's very little oxygen tension? So anyway, that's just a sampling of many of the questions that relate to the physiology and the development of the kernel, for which there's a lot of research that remains to be done.
So you really were changing with time, that you were studying seed proteins for a long time and really changed gears as you went along, which I think is a really important message these days for people starting out in the business. But you also wore many different hats. You were a department chair. You were a PI in the laboratory. You eventually were an associate vice chancellor. You always maintained presence in the scientific literature. And how do you balance those things? And do you recommend that for other scientists?
You left out one thing.
What's that?
I was also a teacher.
Ah.
In fact, during the period of time that I was department head at the University of Arizona, I didn't do very much teaching. But when I stepped down from being department head, I started teaching introductory biology. And one of my recommendations to young assistant professors who are starting their career is some advice that I got when I was a graduate student, and that is teach introductory biology. The big advantage of it is that the subject matter doesn't change a great deal from year to year, so you're not constantly having to revise your notes, number one. Number two, you have a great opportunity to interact with young students at the beginning of their career and get them interested in biology or perhaps even what you're working on. You can attract the really bright ones to start doing research in your laboratory, and then you have an extra pair of hands for a number of years. And besides that, it's just a lot of fun to teach. And a lot of people don't like to teach introductory courses, uh, but I always found it challenging and rewarding. Gives you a chance to follow the students throughout their educational career.
That's probably really good advice. Is there any other advice that you would have for, say, new faculty today who are looking at this rather ominous process of surviving in academia? Any particular advice that you would pass along?
The old-fashioned things of just being curious, particularly read broadly. One of the advantages when I was a graduate student was you had to go to the library and look at different journals to find out what had been published. Now you have electronic screens and you can set up your own keywords, so you can do extremely narrow searches of the areas that you work in. And as a consequence, you sort of miss out on, things that you might never have given a second thought to. The other thing is, it's really important to be comfortable with your level of ignorance. I was told many years ago that everybody is ignorant about something. But the problem is a lot of people are very uncomfortable about showing their ignorance, that is, asking questions about things they don't know. It's something that you need to get over. And finally, you never want to be the smartest person in your lab, because if you do, it really limits your frontiers.
Yeah, no problem there for me.
The final thing that really has changed dramatically since I was an undergraduate and a graduate student, it used to be the case that a lot of people would go into biology because it didn't require a very strong mathematical background. You can do some things like taxonomy, or you could do some things like taxonomy that really were non-mathematical. But that's changed dramatically in the last 10 years. Biology, and for that matter, all science, has become extremely computational. And I don't see how you can be successful in any subject matter, any plant subject matter, if you don't have a good background in statistics and computer science and all the computational things associated with DNA analysis. So that, it just keeps building layer upon layer of the things that you need to know. You on the one hand, you need to be a specialist about something, but you also really need to be a generalist about many things, because as you describe my own career, you start off asking a question, and that question will lead you into other questions, and you'll end up working on things that you never anticipated working on at all.
And does that advice translate well to students? Like, if you were a student coming out today, how would you advise them, especially if they were planning for a career in plant science?
Well, I guess the first thing I would tell them is that you want to think very seriously about what you're getting yourself into here. Uh, doing science is all about discovery and doing research and learning. And if you're not really swept up with the excitement and the rewards that come with learning new things, chances are you won't be really successful at it. The day that you get your PhD is the day you get a hunting license to go out and earn a living, and you have to be willing to put in a lot of time and hard work, and be prepared to fail at things. But the successes are very sweet. I mean, one of the things I told my students is that, I ended up traveling all over the world, and it didn't cost me a dime, because I would be invited to go to meetings or give talks or whatever. And so there's great rewards that come with the profession. There are also great sacrifices that are associated with it. I guess the other piece of advice I would give to undergraduates and graduate students is be very careful about choosing your major professor. They're going to have a bigger impact on your life than your parents did. You didn't get to choose your parents, but you can choose who you're going to do your degree with. So check out what the other graduate students tell you about the faculty before you land on somebody. You're going to be an indentured servant for a number of years, and you want to work for somebody that you know is going to have your best interests at heart.
No, that's really beautiful advice. I think it's something that I certainly agree with. Is there any place that we can learn more about you, or are you active on social media, or have a website that we could refer to?
Well, I haven't done anything. I retired about 6 months ago, so I really haven't had a chance to get anything set up here at the University of Arizona. So right now, all you can do is Google somebody, Google my name, I guess, and see what you find.
Okay, so that's a good start, but dive in on places like Twitter or even Facebook with a professional page. A lot of senior faculty have found it really a rewarding place to be able to engage with younger faculty and students, and really like Dr. Andrew Hansen here at University of Florida. He's been around for a while, but he's really had a renaissance in his connections with postdocs and students, and really providing a lot of very good career guidance that I think is better than any website. So, always it's a great place for folks like you to dive in. I really encourage you to do that. And when you do, let me know, and I'll be happy to put that information out there.
Well, I do have a Facebook account, and I do have a LinkedIn account. I just have never devoted a lot of time to it, at least up to this point.
So, Dr. Brian Larkins, thank you so much for your time today in discussing the book on maize kernel development and also sharing some of your experiences. It was really helpful.
Thank you, Kevin.
And that's the interview with Dr. Brian Larkins for the American Society of Plant Biologists. I'm Kevin Folta. Thank you for listening.